Hello, I'm Dr. Nadia Boutawi. I have a PhD in genetics and an executive MBA in healthcare. I will be talking about SARS-CoV-2 or coronavirus vaccines. In this talk, we'll go over background about the pandemics, vaccine development, leading vaccines and how they work, as well as vaccination challenges. Let's start with some background. In the past 2 centuries, we've seen multiple flu pandemics. The Spanish flu in 1918 killed 15 million people. The Asian flu in the late 50s killed 1.5 million. The Hong Kong flu in the late 60s resulted in 1 million deaths. And in 2009, we had the pandemic flu. We are in the midst of an unprecedented pandemic with over 3 million deaths worldwide. We've experienced healthcare overload and major business closures worldwide. This diagram shows the structure of the COVID-19. It includes the spike protein. That's where it gets the name coronavirus, an RNA, and an envelope. Coronavirus infections are not new. In the 1960s, there were reports of these viruses, SARS in 2002, MERS in 2013. They were present in many countries, with the worst being COVID-19 in 220 countries and territories. The fatalities are also very different, with COVID-19 causing over 3.3 million deaths as of mid-May 2021. Vaccine development is also not new. It started in 1921 with the development of the tuberculosis vaccine. We had the flu A H1N1 in 2009 and Ebola in 2019, with many more before that. As of late 2020, we had the first COVID-19 vaccine. Next, we will review the vaccine development. Vaccine development takes 10 to 15 years in a typical timeline. Each clinical trial phase follows the completion of the prior phase. It can take a long time to accumulate cases to assess the efficacy of the vaccine outside of pandemic conditions, and the manufacturing capacity is scaled up after phase 3 trial and the regulatory approval is completed. In the accelerated timeline, it takes 1 to 2 years. Some clinical trial phases are combined, and because we are in the middle of a pandemic, cases accumulate rapidly to assess the efficacy of the vaccine. The approval is on a rolling basis from early stages of clinical trials, and the manufacturing capacity is scaled up during the trials, but at a great financial risk. All of these conditions resulted in an accelerated timeline, reducing the vaccine development from 10 to 15 years to 1 to 2 years without compromising any steps. Some important characteristics of vaccines include their safety, efficacy, does it work in different target populations, and how long will the protection last? Now let's review the different types of coronavirus vaccines. In the DNA-RNA vaccines, it uses DNA or RNA to teach the immune system to target the viral proteins. In the live attenuated vaccine, a weakened version of the actual virus is used. In the inactivated vaccine, it uses the whole virus that was inactivated by heat or chemicals. In the subunit vaccine, it uses a piece of the virus to focus the immune system on a single target. In the viral vector, it takes a harmless vector to deliver the viral gene to build immunity. All of these technologies have been successfully used to produce vaccines in the past. Now let's review their advantages and disadvantages. In the case of DNA-RNA vaccines, they are easy and quick to design, and that's why they were first to market. In the live attenuated vaccine, it triggers a strong immune response and may not be safe for people with compromised immune system. The inactivated vaccine is safe as the virus is dead, but it's not as effective as the live virus. In the subunit vaccine, it focused response, but it may not stimulate a strong response. Last but not least, the viral vector, it elicits a strong response. However, it's very important to pick a vector that is truly safe. As of mid-May 2021, there are 90 vaccines in clinical trials and 77 more in preclinical animal studies. There is a total of 6 approved for limited use and 8 approved vaccines for full use. Next, we'll review the lead-in vaccines. Lead-in vaccines include mRNA vaccines, such as Moderna, Pfizer, BioNTech, the viral vector, such as Sputnik V, J&J, and Oxford AstraZeneca, the protein subunit by Novavax, and the inactivated virus by Sinopharm. We'll give an example from each of these categories in the next few slides. Let's start with the mRNA vaccines since they were first to market. These mRNA vaccines are not new, and the technology and the science was present and evolving for the past few decades. In the 90s, the first mRNA was injected into mice and produced proteins. In 2000, there was mRNA cancer vaccine trials, and by 2018, there were 8 human trials using mRNA vaccines against HIV, rabies, influenza, and Zika virus. So how do the Moderna mRNA vaccine work? The spike mRNA is packaged in a lipid nanoparticle that protects and delivers these mRNA into the cytoplasm of the target cell, not their nucleus where the genetic code is. These vaccinated cells make the spike protein that in turn triggers the immune response. Sputnik V is a 2-viral vector vaccine. AD26 is used in the first dose, and AD5 vector is used in the second dose to boost immune response. The adenovirus AD26 delivers the COVID DNA to the cell. This vaccinated cell then builds the spike protein, which in turn triggers the immune response. How does the Novavax protein subunit vaccine work? A modified spike DNA is put into a baculovirus to infect a moth cell. These cells make a lot of spike protein. These spike proteins are then extracted, purified, and used to build a spike nanoparticle that is used as the vaccine. Once spotted, it triggers the immune response. In the case of Sinopharm inactivated virus vaccine, the virus is inactivated chemically using beta-propiolactone before vaccination. The vaccinated cells then make the spike protein, and that triggers the immune response. How do the COVID-19 vaccines compare? The vaccine efficacy ranges from 62 to 95% depending on the technology used. Oxford AstraZeneca is the most cost-effective at $3 to $4. It also has the lowest efficacy. Most vaccines require 2 doses except J&J that requires 1 shot. This is important for certain populations where it's harder to administer 2 doses. Sputnik V has a high efficacy and it's cost-effective at $10 per vaccination. They also have one Sputnik-like in the pipeline with one dose only. Sinopharm has 78% efficacy, but it's the most expensive of all available vaccines on the market. In the last section, we'll review the vaccination challenges. Herd immunity is when enough people are vaccinated and it is hard for the disease to spread. This graph shows the overview of vaccination coverage in 2019, which ranges between 40 to 90%. The COVID threshold for infection elimination is between 50 to 80% based on virus reproduction number of 2 to 5. For large-scale vaccination, we need multiple vaccine platforms, coordination for testing, manufacturing at scale, and delivering vaccines equitably. Here are a few challenges for large-scale vaccination: gaining public trust, logistics, human resources, and equity. Early bilateral deals between high-income countries and vaccine developers funded vaccine research and put these countries first in line. For instance, Canada claimed 600% of what it needs, 400% for the US and the UK, and 300% for Australia and the EU. Low-income countries will not get enough vaccines until late 2021 or early 2022. Most vaccines administered to date are in North America, Europe, and Asia. COVAX is an international effort to improve access to vaccines, was established last year. Think of COVAX as the Paris Climate Agreement for vaccines. It is helping somewhat and unlikely to reach vaccine distribution equity in short term due to pre-existing bilateral agreement. High-income countries pledged to give excess vaccines they pre-ordered to low-income countries. In summary, open innovation and collaboration resulted in multiple vaccines in record time. The COVID-19 vaccine development was rigorous, and these vaccines are safe and effective. International coordination is needed to secure vaccines for low to medium income countries, because no one is protected until we are all protected. Thank you for your attention. You can follow me on social media on Facebook at Dr. Nadia Boutawi, on LinkedIn and Twitter, and subscribe to my YouTube channel, Nadia Boutawi. Thank you again.